So, moving on to our second talk of the day, I'd like to welcome Shawnee Traylor, who's going to talk to us about using autonomy to look at the biological carbon pump. So, over to you, Shawnee. Thanks. Shawnee, I'm not getting any sound. Sorry.
How about now? Is that better?
Perfect. You're right. Thank you.
Sorry, I just moved, so I'm figuring out my new setup here. Thank you all for coming and for the wonderful setup from Stevie. My name is Shawnee Traylor. I recently completed my PhD at MIT and the Limnol Oceanographic Institution. Now I'm a postdoc at the Monterey Bay Aquarium Research Institute, where I continue to work with autonomous vehicles to look at carbon cycling in the ocean. I don't have to convince anyone here that the ocean is extremely important in the global carbon cycle. It's the largest active carbon reservoir. It's the dominant natural control in atmospheric CO2 over kind of human relevant timescales. And we owe it a really big thanks for taking up a large portion of our annual CO2 emissions. And this happens through a complex combination of physical, chemical, and biological pathways. One of the primary pathways being the biological carbon pump. So this refers to a web of processes that controls the transport and transformation of carbon from the atmosphere into the surface ocean, and then eventually to the deep ocean, where it can be sequestered for hundreds of thousands of years. It's called a pump because it transfers carbon against that natural gradient in the ocean. And in brief, it's phytoplankton in the euphotic zone, taking up CO2 from the atmosphere and producing the organic matter of which they live and grow. Very small fraction of that primary productivity is then transported to the deep ocean as either generally POC or DOC, where it's out of contact with the atmosphere. So while less than 2% of this carbon is ultimately incorporated to ocean sediments, the BCP does maintain a significant vertical DIC gradient that facilitates CO2 uptake by the surface ocean. And in fact, that's thought to maintain the atmosphere around 200 parts per million lower than with an abiotic ocean. And so the current global estimate for the BCP magnitude ranges between 5 to more than 12 petagrams of carbon per year, which is problematic because that uncertainty range is comparable to the magnitude of anthropogenic fossil fuel emissions. Furthermore, a lot of climate models are usually informed using remote sensing data, which is great. It gives us a huge spatial range, but it cannot resolve a lot of the subsurface processes that are necessary to capture that full complexity of the biological carbon pump. However, it's thought that if we can gain a mechanistic understanding of the BCP, we could potentially be able to predict these subsurface processes using surface characteristics. And that is the goal of the NASA Exports campaign, which is the framework in which this work that I'll be talking about today was completed. So Exports was a large field campaign, or two field campaigns, that was really seeking to kind of throw all of our technology, all of our expertise at this problem, characterized to end member states, and then use what we learned in that to develop better remote sensing algorithms to one day be able to predict really the export fate and carbon cycle impacts on ocean net primary productivity in a changing world. So to gain this understanding, we first required a lot of observations from ships and autonomous platforms. As I mentioned, this project focused on two end member ecosystem states. So the first one being the North Pacific at Ocean Station Papa in the late summer, which is characterized by an iron-limited system with really low export efficiency. Only around 1 to 5% of NPP, or net primary productivity, reaches the mesopelagic, but this is largely zooplankton mediated. And then there's really low eddy kinetic energy, which makes it a really good study site for any sort of Lagrangian or quasi-Lagrangian framework that we would like to be working in. On the other hand, the second cruise was focused on the North Atlantic spring bloom, which is, of course, really highly productive, really high export, very physically dynamic. And this region is extremely important. It represents between thought 10 to 25% of that global biological carbon pump. So I threw this in because these are some knock ships for the North Atlantic leg. I sailed on the Discovery, which was a really wonderful ship. Very grateful to have had that experience. The James Cook was also out there. Anyway, just a cool shot of that. This was the North Atlantic on an uncharacteristically calm and sunny day. And so from these ships, we deployed our autonomous underwater vehicles, which included both gliders, as you see on the left, and floats. Those are two biogeochemical Argo floats. They carried kind of your standard suite of oceanographic sensors with a few more exotic ones, such as the biooptics, pH, and nitrate. I don't know everybody's background, so just a very quick terminology refresh. We'll be talking about gross primary production, or GPP, which is the sum of all the photosynthesis happening. Net primary production, which is GPP minus any autotrophic respiration. And then NCP, which is GPP minus all of the respiratory losses. Now, this metric is really important in the carbon export world because it represents that excess fixed carbon in the system that is available for export. So at a steady state and a long enough timescale, that NCP should equal how much carbon is exported out of the surface ocean. And so for this work, I tried to quantify as many of those values as possible from GPP and NCP to really trying to trace the fate of that NCP autonomously. So one thing that they don't tell you about oceanography before starting grad school is that, well, sometimes work looks like this. Other times it looks like this. These were days of 6 to 7 meter swells. I imagine a lot of you have worked on the North Atlantic and know how that goes. But it turns out that this bad weather was extremely important and impactful to the ecosystems and that carbon export efficiency that we saw. One of the key differences between the two sites was really seeing just how much that physical environment was impacting the biogeochemistry. So on the left-hand side, you have that Ocean Station Papa site, really stratified, very low winds relative to the North Atlantic, where you have a lot of deep mixing. The mixed layer really wasn't stabilizing during the cruise period, which is between those two white dashed lines. It started to stratify actually after site occupation, which again, having the BGC Argo floats out there actually allowed us to continue our observations after the cruise period. But so we see this all reflected in the biogeochemical parameters. So the highly stratified conditions in the North Pacific acted like a lid on the system, which prevented nutrients from mixing up into the euphotic zone, whereas in the North Atlantic, these big storms continually mixed up material up and down and were fertilizing these phytoplankton blooms. And so we see this reflected in the productivity parameters that I discussed before. So on the right, you have GPP and NCP in those two graphs. Traditionally, GPP is measured using bottle samples, which are very time-consuming, require experienced personnel on a ship, and do carry this risk of unknown bottle effects. Whereas with AUVs, we can expand our spatial and temporal monitoring and capture a higher degree of natural variability. And we do this by utilizing high-frequency in situ measurements to capture diurnal fluctuations in oxygen and isolate out the biological signal. And from that, we can calculate the metabolic rate and GPP. And so from this, we found that the North Atlantic site was nearly 5 times more productive than the North Pacific site during that cruise period. And that's all well and good, but again, most of that productivity is just consumed and respired in the surface ocean. And what I really care about is carbon export. So looking at NCP, which I calculated using an oxygen mass bottle or mass balance model, we found that in the North Pacific, the system was lightly autotrophic. Nutrients such as silicate and nitrate were abundant, which again supported this idea that iron was limiting. Small blooms would grow and then collapse, but the system was largely recycled and not much carbon escapes to the deep. Meanwhile, in the North Atlantic, NCP was about 12 times that of in the North Pacific. It's very highly autotrophic and had really high amplitude swings. And we seem to have arrived on the tail end of a previous diatom bloom and the second half of a kind of dual phase bloom that has been described before, such as in the 2008 North Atlantic bloom experiment. So the big picture, we found what we were hoping to find. We found these two really contrasting end members of productivity and export regimes, and we did the whole thing autonomously. Great. North Atlantic was dramatically more productive. Higher percentage of that carbon is being routed to NCP, presumably to be exported from the system. And I can show tables and graphs that validate these methods against the climatology and show you that I think we did a decent job at getting these numbers right. But there is this caveat that I mentioned earlier that I think you should hold me accountable to. And that's not an equal sign. And I never really defined what a long time is. As we all know, the ocean is a highly dynamic place, and a steady state assumption doesn't really apply to many regions or timescales of observation. That new organic material that's produced and reflected in NCP might not all sink out. Some, of course, remain suspended in the water column. For example, though observations at the North Atlantic field site continued after the cruise period using those BGC Argo floats, that analysis could only be extended by an additional 3 weeks before the cohesive eddy structure in which we were working dissipated. So numerous approaches to this challenging measurement of carbon export have been employed, from sediment traps, optical traps, isotopic tracer analyses, and they all have their advantages and shortcomings. Additionally, they all measure the particulate side, which our oxygen-based approach does not. And so we're really thinking about like, well, how can we, leverage these new technologies that we have on the AUVs to come up with some sort of measure of carbon export that could be done autonomously. There certainly are, gold standards of POC flux measurements, and we are not trying to replace that, but the spatial temporal coverage that we can get with an AUV is dramatically different. And so thinking about this, we kind of came up with, well, we can look at that. We can break down the change in the POC inventory as some of the balance between production, removal, and any sort of advective processes. And the NCP that I calculated before is one of these terms, and we can kind of rearrange this equation to solve for it. And while we can't measure all of these terms autonomously, we can leverage those biooptical sensors on the AUVs to measure the POC, the particulate organic carbon, and define a new term, this organic carbon flux, that equals NCP, but is corrected for that kind of suspended particulate carbon that might be remain in the surface ocean during the period of observation. And that in turn equals the net production of DOC plus the removal of particles via diel vertical migration and gravitational sinking, which are those latter two terms. This correction was a lot more important for the North Pacific site, where the magnitude of POC inventory change and NCP are similar magnitudes. Furthermore, we can take advantage of the fact that thorium-based POC flux measurements were taken during the cruise, which, again, unlike the method we're employing here, only captures removal from the particulate pool, whereas we are seeing both dissolved and particulate carbon pools. And so that implies that the difference between those two terms can tell us something about net DOC production. Importantly, we can't differentiate between net production, accumulation, and export, which are going to depend on the vertical and lateral movement of water masses. And I can't say anything about production mechanism, but we can have some sense of maybe how important DOC production might be when trying to balance our carbon budget. And so the organic carbon flux was 6 times higher in the North Atlantic than in the North Pacific. In the North Pacific, there were small export fluxes and the flux terms were approximately equal. So this is really suggesting that organic matter is primarily exported through those particulate pathways, whether it's gravitational sinking or zooplankton mediated, such as diel vertical migration, with no significant DOC production, which is in agreement with Huang et al. From 2022, an independent study of a similar site. Whereas in the North Atlantic, NCP is being routed to near equal proportions of DOC and POC, particularly at those shallower integration depths. Down at 100 meters, NCP and the thorium-based POC flux are in balance. That suggests a nearly equal amount of NCP in the upper 100 meters is exported as POC during the cruise period. And it's important to keep in mind that, we're always working across slightly different integration timescales. So the thorium flux can be the thorium-based POC flux can be capturing earlier productivity that is being exported during this time of site occupation. So a quick note about export ratios, which are a really handy way for us to kind of compare sites. So the EF ratio is that ratio of NCP to NPP and reflects the fraction of organic matter that's fixed by productivity that persists at a given depth horizon, whereas the EZ ratio is sinking POC flux to NPP. That really is getting an export from the euphotic zone. And so these are the values for both of these calculated from the autonomous values. So instead of the POC flux here, I'm using that organic flux term. And this is just to say that if we look back at those kind of the earlier graphic I showed, that really informed the choice of these end member sites that was based on the EZ ratio and this flux transmission, can recalculate these values and see how they look. So it looks like our estimate is an upper bound. So if you're in a site where no POC flux is available, if no data are available, this could be a useful tool. I think if we're in a site that's known to have where DOC production is known to play an important role, it's less useful. So in the North Atlantic, we'd be way, way overestimating relative to previous studies. Whereas at Ocean Station Papa, there is a slight overestimate, that could also potentially be real variability across the years. But to summarize, so we were able to demonstrate how we can use sensor-based observations to track carbon metabolism all the way from photosynthesis in those GPP parameters to export from NCP to really trying to get at some sort of particulate flux autonomously. And this is really providing a lot of insight into how the biological carbon pump is working in a particular area. We were able to show within the context of that exports program that we captured these ecosystem end members of export efficiency. As I said before, we all expected the North Atlantic was much more productive than the North Pacific, and had greater export efficiency, which was reflected in those kind of typical EF ratios, EZ ratios, and the total cruise integrated organic carbon flux. And finally, we were able to tease out a bit how NCP was being routed to the DOC and POC pools in different proportions at each site, which I think is it can be a really useful tool if we can even look back through historical data and see where we might have the ability to compare or to leverage existing autonomous vehicle data sets to perhaps POC flex work that has already been done. I think there are a lot more analyses that could be done with this kind of combination of in situ and autonomous based work. Finally, there are a lot of hands in this pie. So thank you to everyone that was part of this project, all of our funders, and thank you for your time and attention. I'm happy to take any questions.
Thank you very much, Shawnee. Another great talk. Thank you very much. So once again, open to the audience for any questions or comments. Let me just scroll down, see if there are any hands going up. So I've got one. This is sort of an impossible question. I should warn you in advance. So I think you've done a really nice job of describing where we are with autonomy, in that it's opening up some fantastic avenues, but we're still a little limited in terms of the sensors that we have available. So you also sort of showed us that equation where you've broken down sort of the creation, sort of destruction of organic material. So if in a perfect world you could conjure up a sensor for just one of those terms, which one would you pick on? Which one do you regard as being the one that is sort of the bane of your life when you're trying to do these calculations?
Oh, that's a really good question. For export flux in particular, well, I'll fully admit there's a lot that can be done with bioptical sensors that I'm not super familiar with. I'm very far from an expert in that. I come from a dissolved gas background. But there are limitations. Like I was using backscatter at 700 nanometers. That is going to capture certain particle sizes and not others. So I think if there were some, and I know people are definitely working on this, people with much more expertise in it than me. But I think these kind of like optical based particle sensors have some really promising things coming out of them, and people are using them in very clever ways as well to really tease out, particle size distributions. And I think that will open up a lot of avenues even within the modeling world potentially because of the differences in, gravitational sinking rates and things like that. I think that this is a very exciting kind of avenue. So I guess I would say if we could really nail what those particles are, how big they are, I think that would open up a lot of avenues for really being able to trace them, laterally as well, which one thing I wasn't explicit about is that at both of these study sites, we were operating within an area, whether it was a mesoscale eddy in the North Atlantic or just a really low eddy kinetic energy area in the North Pacific. We were trying to minimize any sort of horizontal fluxes, which is less of an issue for oxygen, which again is most of my work in the surface ocean thing. The horizontal gradients are relatively small. Thanks for the question.
No, at all. No, great answer. Thank you. Francisco.
Oh, hi again. Thanks, Shawnee, for the talk. Another excellent talk. My question goes to the same. I mean, it's about the same equation that Adrian was inquiring. It's about the net advection term. How large is that in your? If you have computed it separately, how large was that term compared to the others?
Yeah, thanks for the question. I did compute it. I don't have a graph or anything prepared. It was not huge. As I said, we tried to operationally design the studies in a way where we could be minimizing those effective fluxes. I do think that, these are all parameterizations. You do the best you can. I think that is probably one of the weaker points for us is really being able to accurately know what those lateral movements are. Several of my co-authors are physical oceanographers, so I kind of accept the numbers that they gave me, but I guess relative to the other terms, they were small. And I can drop the link to the paper in the chat where there's more detail.
Oh, no, thank you.
Thank you. Yeah, Shawnee, if your co-workers include Eric and Craig, then I think we'll take your word for it.
I was taking their word for it.
Yeah, absolutely. Yeah, you're not going to find me arguing. But, excellent. We've still got time for one last question if people want it. Otherwise, we can wrap things up. Let me just quickly go through the list of things just in case anybody's got one more question. I should just check. No, so I think there we have it. Once again, many thanks to Shawnee, but also to Stevie for 2 excellent talks. I'll try and get the talks uploaded to the YouTube if you want to watch them again. Answer any questions you might have. Please do feel free to contact the 2 speakers. I'm sure they'll be delighted to hear from you and answer any questions. And keep an eye open for the next call and talks, which will hopefully be advertised before long. So thank you all very much, and thank you for joining.